Let me give you another example of irreducible complexity. I want to talk to you about bacterial cell division briefly. So here you can see a photograph, if you will, of a bacterial cell as it's dividing. And you can see this is called the division septum. This is where a group of proteins that are called FITZ, don't worry if you don't remember the name, but they're called FITZ, and they basically polymerize or assemble along this division septum. And these bacterial cells elongate and basically they bifurcate into two daughter cells. And so that's how bacterial cells divide. But as you might be expecting, it's not quite as simple as that. I'm not going to go through all this because it's a little more technical than I'd like to get into, but this is some of the essential proteins of the bacterial divisome. I want to focus particularly on this is, as I mentioned, this is FITZ here, this ring structure which forms around the point of division. And FITZ is essential across bacteria. It's absolutely essential. You knock FITZ out, cells don't divide. But FITZ has to be tethered. It has to be tied to the membrane. And so you have proteins involved called ZIP-A and FITZ-A, which tether the FITZ proteins to the membrane. And if you knock out the tether proteins, again, the cell can't divide. And so this is what you might call an irreducible complex system. But as I'm about to show, it actually gets a lot worse than that. Bacterial cell division by binary fission requires the growth of the peptidoglycan cell wall as the bacterium elongates. This process occurs by the severing of the peptidoglycan backbone and the synthesis of new cell wall material. Now, there are enzymes that are called autolysins, which basically hydrolyze the subunits that form the peptidoglycan cell wall, and the gaps are filled in with additional cell wall material. But since these enzymes can result in programmed cell death, they require careful regulation. And this is just a process. This is just an illustration showing the process by which the cell wall is constructed. It's called transpeptidation, and the subunits that form the peptidoglycan cell wall have to be brought in by they have to be brought in by a protein called bacterial, which bonds to these precursors of the peptidoglycan and transports them across the cytoplasmic membrane. So that's required as well. Now, I want you to consider this evolutionary enigma. So consider the two following observations. Number one, critical to the elongation process is the severing of the peptidoglycan cell wall by the autolysins. All right? Secondly, critical to cell viability is the resynthesis of the peptidoglycan cell wall. Breaking the cell wall can serve no adaptive utility until a mechanism has arisen for the simultaneous integration of peptidoglycan precursors. Indeed, without the latter mechanism, the cell is rendered non-viable. So you have an irreducible complex system, because in order to elongate, you have to break and sever the cell wall, right? But if you don't rebuild the cell is non-viable. So you have to have the mechanisms together, and so that's an irreducibly complex system. That's a tremendous challenge to the evolutionary hypothesis.